Today we will take restriction enzymes. You are all well aware of enzymes. You know that enzymes are biocatalysts which speed up any biochemical reaction. For example, you know that enzymes like trypsin, pepsin, amylase, lipase, all these are digestive enzymes which help in the digestion of food material. But here we are taking a special enzyme called restriction enzyme. These enzymes, restriction enzymes, are nucleic acid digesting enzymes. They digest the double-stranded DNA, and their importance is that in spite of being enzyme, their activities are quite restricted. Restricted in the sense that they require special nucleoside sequences for their activity. Special nucleoside sequences means they recognize a specific recognition sequence, means a sequence of nucleotides. If they are present, then these enzymes will show their activity. These restriction enzymes will show their activity, and if those recognition sequences are absent from the DNA molecule, then they do not show any activity. That's why they are termed as restriction enzymes. They are also termed as molecular seizures. Molecular, you know that a molecule is a very small entity, and seizures, you know, in Hindi we call it as कैंची. Just like एक कैंची की जरूरत पड़ती है यदि किसी कपड़े की कटिंग करनी है, similarly in the recombinant DNA technology we have to cut the DNA molecules and then ligate them together, and that cutting and joining is held by these restriction enzymes. That's why these restriction enzymes are the key component of the recombinant DNA technology. If you successfully understand the mechanism of action of these restriction enzymes, then you can easily understand the recombinant DNA technology. Here in the diagram you are seeing that there is a double-stranded DNA and it is cut by a restriction enzyme. Since we cannot see the DNA molecule by our eyes, हम अब कपड़े को तो देख सकते हैं जिसको हम कैंची से काटते हैं, but we cannot see the DNA molecule. So the instrument required to cutting this molecule is also is invisible. वो भी नहीं दिखाई पड़ता. That is restriction enzyme. But since this restriction enzyme has a very precise role and it recognizes a very specific DNA sequences, so we can easily detect that what is the sequence over which this restriction enzyme is cutting, and finally what will be the result of that particular sequence of DNA, or you can say DNA fragment. Now, first of all, we will take the definition of this restriction enzyme. Restriction enzyme can be defined as restriction enzyme is also termed as restrictase. They are naturally occurring endonucleases. Endonucleases means they cut the DNA sequences from in between the DNA strand. Their activity is not at the two ends of the DNA molecule, but in fact they cut in between the two ends. That's why they are termed as endonuclease. So restriction enzymes or restrictase are naturally occurring endonucleases that recognize a sequence of specific bases of DNA called recognition site. Means the sequences which are required for their activity are termed as recognition sites, and they cleave the double-stranded DNA into fragments when they recognize that recognition site. These enzymes are found mainly in bacteria and archaea, means in prokaryotes, and provide a defense mechanism against the invading viruses. We will take this explanation in certain other slide. Now, the recognition site or restriction site is the site which is constituted by about 4, 6 or 8 nucleotide bases. 4, 6, 8 nucleotides. Generally, they are composed of either 4 nucleotides or 6 nucleotides. Rarely, 8 nucleotides may also function as restriction site or as recognition site. An interesting feature of these restriction nucleases is that they recognize a DNA sequence that are palindrome. Means these sequences, either 4 or 6 or 8 bases, are palindromes. Palindromes means the sequences that read the same in forward direction as well as in backward direction from the central axis of the symmetry. Means from certain point, if we read them from this side or from the back side, in both the directions they will mean the same thing. For example, suppose that there is a sequence from the 5' end G A A T T C, and this is the 3' end. Now the other strand will have 3' C T T A and G. Five dash. Here you see that from this line there is a point of symmetry. Here you see that GAA 5' prime, GAA TTC, and again back you read both the sequences are exactly similar. Such sequences are termed as palindromic sequences. Now, there are many restriction enzymes which has been recognized so far from different prokaryotic organisms, and for example here you can say HPA second that has been recognized from Haemophilus parainfluenzae. The recognition sequence is composed of this G, C, G, G. Similarly, there is eco-R1 that has been detected in Escherichia coli R13. eco-R2 means second enzyme which has been separated out from Escherichia coli from the same strain RY13. BAM H1 Bacillus amyloliquefacius. Now we will take where from these restriction enzymes and why these restriction enzymes evolved. Restriction enzymes evolved to protect the bacteria from foreign DNA. Foreign DNA means generally bacteria are infected by these viral invaders, means viruses, bacteriophages, and they take over the bacterial DNA, and destroy the bacteria, and they survive themselves on the food materials and the biochemical materials which are present in the bacteria. So to protect themselves from these foreign invaders, that is bacteriophages, for example, just to protect from foreign organisms, our body respond by our immunity. For example, if any antigen enters in our body, then these antigens are recognized by our immune system, and accordingly antibodies are produced which finally remove these antigens. Similarly, whenever these bacteriophages enter inside the bacteria, then they are destroyed by these restriction enzymes. These enzymes does not cut. Now, how these enzymes act? How they specifically destroy the viral genome, but there is no effect on the bacterial genome. This is possible because these enzymes are methylation sensitive. Methylation sensitive. These enzymes are methylation sensitive, means they recognize the restriction site or recognition sequences only if those sequences are not methylated. If they are methylated, then these restriction enzymes cannot detect those restriction sequences, and if they are not recognized by the restriction enzymes, then they do not cleave at those locations. And how this is possible? In fact, a bacterium produces a modification enzyme. With the restriction enzyme, always a modification enzyme is also associated. These modification enzymes recognize the same sequences as the corresponding restriction enzymes. Means जैसा जो sequences restriction enzyme determine करता है, उसी सेम सीक्वेंस को ये मॉडिफिकेशन एंजाइम भी डिटरमाइन करता है, और ज्यों ही उसका पता लग जाता है कि ये सीक्वेंस बैक्टीरियल जीनोम में प्रेजेंट है, एट द सेम टाइम दिस मॉडिफिकेशन एंजाइम मिथाइलेट ईच ऑफ दीज रिकॉग्निशन साइट्स इन द बैक्टीरियल जीनोम, और जैसे ही ये मिथाइलेशन हो जाएगा उस रिकॉग्निशन सीक्वेंस का, now those sequences cannot be determined by the restriction enzyme, and as a result restriction enzyme will not be able to cleave those sequences. Generally, the sequences which are methylated, the nucleotides which are methylated are either adenosine or cytosine. Once the DNA is methylated, it is protected from the restriction endonucleases, और इस तरीके से बैक्टीरिया के पूरा का पूरा जीनोम वो जो भी रिस्ट्रिक्शन एंजाइम प्रोड्यूस करता है उसके अकॉर्डिंगली मॉडिफिकेशन एंजाइम उन रिस्ट्रिक्शन सीक्वेंसेस को वो मिथाइलेट कर देता है. एज ए रिजल्ट दोज सीक्वेंसेस कैन नॉट बी डिटरमाइंड बाय द रिस्ट्रिक्शन एंडोन्यूक्लियेजेस. एज ए रिजल्ट दीज रिस्ट्रिक्शन एंडोन्यूक्लियेजेस और रिस्ट्रिक्शन एंजाइम्स विल नॉट बी effective against those sequences, means they could not be destroyed by the restriction enzyme. The entire DNA in the bacterial cell, including the chromosomes and any plasmid, is normally the hair is protected by these modification enzymes. इस तरीके से ये restriction enzymes फॉरेन इन्वेडर्स के लिए, वायरल जीनोम्स के लिए तो डिटरमाइन करके उनको डिस्ट्रॉय कर देता है, लेकिन अपने ही होस्ट जीनोम को मीन्स बैक्टीरियल जीनोम को या उसमें प्लाज्मिड जो प्रेजेंट है उसमें चूंकि वो सारे सीक्वेंसेस मॉडिफिकेशन एंजाइम के द्वारा मिथाइलेट कर दिए जाते हैं, इसलिए दोज रिकॉग्निशन सीक्वेंसेस कैन नॉट बी डिटेक्टेड. एज ए रिजल्ट द रिस्ट्रिक्शन एंजाइम विल नॉट एक्ट ओवर दोज सीक्वेंसेस. Now, अब हम देखेंगे कि हाउ these modification enzymes in fact act. Suppose there is a restriction enzyme eco-R1. Now this eco-R1 cuts at this location, and at this location if a nucleotide sequence G A A T T C is present. Now what does the modification enzyme in fact determines the same nucleotide sequence G A A T T C, and as soon as it detects, it modifies the DNA sequence. For example, here it modifies the adenosine. Again, here the modifies adenosine. It adds a methyl group here and a methyl group here. As a result, now the restriction enzyme cannot detect this GATT sequence, and so the restriction enzyme will be ineffective. तो इस तरीके से अपने होस्ट जीनोम को से रिस्ट्रिक्शन एंजाइम के द्वारा डाइजेस्ट नहीं किया जा सकता, और जबकि जब ये कोई फॉरेन डीएनए इन्वाइट करता है उसमें यदि ये सीक्वेंसेस मॉडिफाइड नहीं है, मिथाइलेटेड नहीं है, then the restriction enzyme will destroy those DNA sequences. Now, how the name of these restriction enzymes are allotted? क्योंकि अब तक कई सौ रिस्ट्रिक्शन एंजाइम्स डिटेक्ट किए जा चुके हैं, तो नेम के लिए, उनकी नेमिंग के लिए जो नेमिंग पैटर्न है वो रिस्ट्रिक्शन एंजाइम्स हैव नेम्स डिराइव्ड फ्रॉम द फर्स्ट लेटर ऑफ द जीनस. मीन्स जिस ऑर्गेनिज्म से रिस्ट्रिक्शन एंजाइम डिराइव किया जा रहा है, निकाला जा रहा है, उस एंजाइम, उस ऑर्गेनिज्म के जीनस का पहला लेटर और उसके स्पीसीज के 2 लेटर्स के द्वारा ये रिस्ट्रिक्शन एंजाइम का नाम रखा जा सकता है. सपोज कोई एंजाइम एस्क्रिचिया कोलाई से निकाला जा रहा है, तो इसके जीनस का पहला लेटर है E, that is here it is written E, और स्पीसीज में फर्स्ट 2 लेटर्स हैं CO कोलाई में, तो इस तरीके से E और CO मिला करके E को नाम हो गया रिस्ट्रिक्शन एंजाइम का. नाउ यदि आगे कोई स्ट्रेन भी है एस्क्रिचिया कोलाई का, देन दैट नेम ऑफ द स्ट्रेन हियर, फॉर एग्जांपल R, here R word is added after eco. So now the enzyme which has been taken out from the Escherichia coli R13 strain is eco R, and if there is a possibility that many more enzymes may be present in the same organism. In that case, the first enzyme which has been determined in that organism is termed as 1, and if any other enzyme is further determined, then 2, 3 in this way. So here the first enzyme which has been detected in the Escherichia coli, its name will be eco R1. So in this way the name of different enzymes are allotted. If the 2 restriction enzymes are from different species, means दो रिस्ट्रिक्शन एंजाइम 2 अलग अलग स्पीसीज से, ऑर्गेनिज्म से यदि मिलते हैं, and both these restriction enzymes act on the same recognition sequence, उसी रिकॉग्निशन सीक्वेंस को पे ही एक्ट करते हैं, उनका रिस्ट्रिक्शन साइट सेम है, देन सच एंजाइम्स आर टर्म्ड एज आइसोसेजिमर्स. Now Once the restriction enzymes has been named, the next thing is describe the different types of restriction enzymes.